Hello everyone, welcome to this webinar on advancing 3-day sterility testing for cell and gene therapies. My name is Per Lidén, Marketing Manager at Symcel, with home base at the Symcel head office at the Karolinska Institute premises in Stockholm, Sweden. Presenting today will be Wilhelm Paulander, head of our clinical development team. There will be a Q&A session in the end, so if you have questions, please ask these using the Q&A function. If there are any hiccups using the function and you're connected with the browser on the phone, please try connecting with the app instead. The webinar is recorded and there will be a send out of the recording after the presentation. With that, I hand over to you, Wilhelm.
Thank you, Per, for the introduction, and good afternoon and maybe good morning to some of you out there. Let's look at the agenda for today's presentation. I will start with going over the calScreener Plus, our platform for sterility testing, look at the setup and the workflow. We will look at the measurement principles and go over some of challenging organisms and see how they perform in our system. We will also look at data from an ongoing primary validation, look at some products and product matrix effects, and specifically then the impact on time to detection, and then finish up with a conclusion and, as Per said, some questions that we can hopefully discuss the technology in more depth and answer some of the thoughts that you had from the presentation. If we start with looking at Symcel and just have a quick overview, as Per said, we're located in Stockholm, Sweden, where our headquarters is, but we also have a site in US, Boston. We're about 40 employees at Symcel at the moment, with quite a large number of consultants on top of that as well. And our main focus is to develop isothermal microcalorimetry for rapid growth-based sterility testing. And this is what we will go over today, to look at our development and see how far we got with it. If you look to the right, you see the calScreener Plus system. It's a benchtop size system with a quite small footprint, as you can see from the dimensions. But to the left of the system, you see 5 points. And if it's only one thing that you take with you from today's presentation, I hope it's these 5 points. And I think it summarizes very well the technology and what the technology can offer. It can give a result within 3 days, so we can give a time to negative within 3 days. The method itself on the sample is non-destructive, so it means that it totally passively measures the sample during the incubation time. And after the incubation is done and you have detected, for instance, a contaminant, you can do any type of the identification. Could be with 16S RNA, full genome sequencing, or MALDI, for instance. Technology is not an endpoint assay. It is a continuous monitoring, measuring every 5 seconds. So you get an alarm as soon as you have something detected with the technology. It's extremely suitable for direct inoculation of product, and I will go over both with examples and explain why the technology is that suitable for direct inoculation. But for instance, we run a lot of clinical samples in other IVD development projects where it's been tissues that are just been homogenized. It could be bones, or it could be, for instance, a T cell. The product itself does not inhibit the signal. And it's a phenotypic measurement. So we only detect living organisms, and we don't detect things such as genomic residues or other molecular markers. If we look at the system and an overview of how the system looks like, it's a twin system with 2 temperatures. One high temperature at 35 and one low temperature at 25. The system can be set to anywhere between 20 and 40, so it's up to the user's discretion what temperature they want to run. We are using 2 different growth media, TSB and FDM, so the classical compendial growth media, but we're using it with one sample in TSP at 25 degrees and two samples, both TSP and FDM, at the high temperature incubation. The sample is introduced via a needle into the vial through a septa. We can take up to 250 microliter of sample per vial. And the minimum amount then, or samples that you run, are 3, because you need to cover all 3 temperatures, but the volume can vary and be split up in between these 3 vials. You can also add vials into the equation in order to increase the amount of sample that you want to test on. In this setup and the data that I will show you today, growth media is also added to the vial. So you have to add 2 things to the vial, both your product and the growth media. But we are developing a pre-filled vial where the media is already in the sample and where you only need to add the product to the sample. We meet the expected regulatory requirements with 21 CFR Part 11, software support, IQ, OQ, PQ protocols, and all the things that are expected from a platform like ours for sterility measurements. We can measure on 32 samples in each unit at the same time. If we then look at the biocalorimetry principle a little bit more in detail. So what we're using is isothermal microcalorimetry. And what it does is that it measures the metabolic activity from any living microorganism. And it does so by measuring the heat produced during the metabolic activity. Any living microorganism or any metabolic process produces heat. It just produces heat to different amounts. We do this by measuring the temperature in the heat detector and then having a heat sink that carries away the heat generated from the sample so we can measure in real time what's produced in the sample. Over time, microorganisms multiply and generate more heat. That, of course, generates a higher signal, but it's not only related to the CFU or the biomass that is generated in the sample. Different microorganisms have different capacity to produce different metabolic output. And we will look into this a little bit in the coming slides, because it both can explain a little bit why we can be so fast of detecting, for instance, slow-growing anaerobic organisms, for instance. It's worth noting that the detection of microorganism is not solely dependent on biomass, as I said in another slide, but the metabolic capacity from each cell. And biomass formation does not mean a high metabolic activity, and vice versa. It can be that a low biomass can produce a high metabolic activity. And if we look at some of these examples of this to illustrate it a little bit more, I think E. coli is an interesting example. It's a bacteria that is facultative anaerobe. It has a short generation time, and it produces a lot of metabolic activity. It has the capacity of both aerobic metabolic activity, using primarily the TCA cycle, but also can switch when it runs out of oxygen into a fermentation metabolic or a anaerobic metabolic activity. And fermentation produces a lot of heat, so it actually accelerates the metabolic capacity even further, or the heat produced during the metabolic capacity. Another example of a fast-growing microorganism with a short generation time is Micrococcus luteus. It has a very small genome and encodes very few metabolic pathways, and also produces little metabolic output. One of the reasons for that is that it can't grow anaerobically and can't ferment. Also, it likes a glucokinase, and it has to drive other things such as not glucose directly into the TCA cycle, such as amino acids and peptides. This produces very little heat and is a tightly regulated metabolic mechanism. So Micrococcus, for instance, is an organism that grows fast, creates a lot of biomass, but produces quite low metabolic output per cell. As a third example, we can look at a slow-growing organism with a long generation time, but that produces a lot of metabolic activity. And I think C. acnes is representative for a lot of slow-growing anaerobic organisms that are relying primarily on fermentation. The advantage, though, with relying primarily on fermentation is that it produces a lot of metabolic activity, and hence it's easy for us or for a system like ours to detect. So C. acnes, even though it's slow growing, can be detected very fast in a system like ours. If we then look into two other organisms that I mentioned, I will start with Micrococcus and then move into C. acnes, because I think they're important and illustrate some of the pros and cons with a system like ours. We can look at the setup of the vials. So I said in a slide earlier, that's a passive measurement. And that means also that the vials are standing completely still because that is needed for the setup of the platform. That means that diffusion of gases and other things are quite low. We don't have a shaking platform, for instance, which is commonly used in order to dissolve gases such as oxygen into the growth environment of the microorganisms. That means that we have had to optimize quite a bit in order to increase the performance of the system. And that was one of the reasons why we added a third vial in TSP in order to optimize the growth capacity and the metabolic capacity of such organisms such as Micrococcus luteus. On the slide, there are three figures, and if you start from the left, we can see the effect of optimization and using different growth media vendors. And we can see that depending on what growth media you can have a very large effect on the metabolic output, so the signal that we are measuring. And we are looking at this as heat flow, and we're measuring it in microwatts. So that's the output or the metabolic output that we measure from the microorganism. If you then look at the middle graph, you can look at the heat flow and what we detect, and also look at it from different temperatures. So if you incubate at 25 versus 35, you delay the time to detection with approximately 20 hours. So by increasing the temperature, you can detect the same microorganisms relatively faster. We also, at a higher temperature, increase the output of the microorganisms, so increase the metabolic activity of Micrococcus luteus. So to the furthest right, you can see the representation of plus a minus product and the growth of Micrococcus, or the metabolic output of Micrococcus, I should say. So a lot of the process that we've done is to optimize the growth media, optimize the growth conditions in order to produce as much metabolic output from hard to detect or hard to grow microorganisms such as Micrococcus luteus. If we then take an organism at the other end of the spectrum, that is relatively easy to detect in a system like ours. And I think it's also interesting from the point of view that it's often a rate-limiting organism in most other growth-based assays. And that is the anaerobe organism C. acnes. C. acnes, as said before, has a very long generation time and often can take everything from 14 days and upwards, even in the compendial assay, to detect. We can detect it very fast, and we can represent it here also by looking into not only the ATCC isolate that you see to the left. Let's see here if I can get a laser pointer that you see here to the left, but we also have plotted quite a lot of clinical isolate from our freezers that we have collected over the years. And I think this picture is interesting from a lot of perspectives. We're not looking at a ATCC isolate, the standard one that you look at, but also looking at how does our system perform when we're running other types of isolates. We have also plotted the detection times of the systems using the detection formula or the buffer time recommended by USP 72 and 73, where you roughly add 3.3 generation plus the generation time of those generations in order to say how much buffer time do you need to add to your assay in order to detect the most slow-growing organisms. And what you can see here is that it's represented that we have a detection within 72 hours for all of these isolates that are represented here. So with these two examples, I think one can look at both a fast growing that produces not that high metabolic activity, that produces a lot of biomass, such as Micrococcus luteus, that we might detect a bit slower than you would anticipate just purely based on growth rate, and then look at a rate-limiting organism such as C. acnes, which you would anticipate to detect maybe within 7 to 14 days. However, we can detect it under 3 days in our system. If we move on to the primary validation data, that I think illustrates very nicely how the system's been performing when testing very defined parameters. We will go through robustness and ruggedness, specificity, and limit of detection. Just to give you an overview, I will walk through some of the robustness and ruggedness parameters. Different organisms has been chosen to be combined with the different measurement points in order to test the parameters to its full extent. So for instance, if something is limiting the amount of oxygen or headspace in the vial, we take in an organism that is sensitive towards a limited oxygen amount. Or if something that is delaying the incubation, we've taken something that is slow growing in order to see how does that affect the detection of that organism. In total, we have run over 1,500 tests for the primary validation, and I think there's a lot of interesting data that we can walk through here. One question that I think is extremely important and that showcases a little bit how the system is working is the impact of the sample volume on the output for instance, oxygen-sensitive microorganisms such as Aspergillus brasiliensis. To the left, we can look at 3 different volumes, 400 microliters, 500 microliters, and 600 microliters. And we can see that there's no significant difference in time to detection. However, when you look at the metabolic output of the organisms, you can see that there's a clear impact when you increase the volume on the metabolic output. This, I think, is very interesting and also important because it showcases how much product you can load in our vials before you might have an effect on either time to detection or that you might actually get a too low signal that would create a false negative in our system. If we then take a further look and look at reproducibility among instruments and operators, this is supposed to be a quite non-exciting slide or even boring slide, and that is good because we don't want any differences here. I think that there's no significant difference in between the different instruments that we tested and no significant difference when you use different operators to test this. We are using 30 replicates for each instrument and for each operator, and that's sort of the standard when we do the primary validation. And in most settings, we use 20 CFU or below. If we then look at the effect of sample insertion timing, so look at the effect on if you don't insert the sample directly into the measurement chamber, how will that affect the time to detection, or will it affect the whole assay? As you can see here, that if you delay the insertion with 6 hours, you get actually faster detection time inside the system, which is maybe not that strange because the microorganism can, of course, divide in the room temperature. However, even though it's a significant result, it doesn't affect our time to detection in total, right? So if we have a 72-hour assay, this doesn't negatively affect or affect the total assay. If we look at reproducibility across media lots, which is an important factor for the stability of the system, We can see that there are differences among media lots, both for TSP and FDM. However, none of the differences, even though significant, impacts the time to detection, or when we call this time to negative for the assay. So we have minor differences in between lots that are significant, but does not have a major effect on the assay in total. If we look at the impact of growth media dilution, if you run 1 times or lower times concentration of TSP and FDM, and this is also to see how does a difference in test sample affect or product sample affect when you add to the system. We can see here, both for Candida albicans and Micrococcus luteus, that there is an effect for Candida albicans that we don't see for Micrococcus luteus. But none of the effects are affecting the assay overall. So even though you see a significant difference between half-time and full-time concentrated growth media, it's not something that affects the end result of our assay. If you go further and look at specificity and limit of detection parameters, we have tried for specificity to cover quite a wide range of different metabolic mechanisms. So looking both at fungi, molds, and different type of anaerobes, and also, of course, challenging isolates that are isolates of concern, for instance. But also not only looking at the kind of classical organisms to add to a specificity assay, but also trying to cover the different metabolic subtypes. For the LOD, that is still ongoing, but almost finished, we have done a LOD and a comparability assay at the same time. We are going to do it for 9 organisms. I will show you 8 today. We are following USP 1223 and European Pharmacopoeia 516. And the method that we're using is quite standard, but it's the most probable number estimates, where we're comparing our platform, the calScreener Plus, with the compendial method in FDM and TSP. And we are classifying it as non-inferiority if there's overlapping 95% confidential intervals in between the calScreener Plus and the compendial method for the most probable number estimates. If you look at the specificity data, it's divided in 3 different temperatures. If you look at the organisms, some of the organisms you might be expecting in FDM, it would maybe rather be in TSP or 35 or 25, for instance, here. Or there might be something that you usually standard grow in TSP 25, but we have it in TSP 35. In most cases, if they grow in FDM, and the standard grow also in TSP and 25 and 35, they will grow in our system as well. So basically, whatever you get in growth in the compendial system, you will get in ours as well. But time to detection can vary since we're looking at the metabolic output and not the biomass formation. And this is really what we're showing here. So even if they biomass-wise could be growing faster in another setting, we are really displaying where they are detected first metabolically in our growth media. And as you see here, for the organisms that we tested, they're in between 5 to 20 CFU. We have 30 replicates per tested organism, and we get a very nice performance, I think, of all tested organisms so far. And rate-limiting organism in our system has been C. acnes in all cases, and I showed you data before there where we are below 5 CFU for C. acnes isolates. If you take a step into limit of detection, we have done it with 10 CFUs, 1 CFU, and 0.1 CFU as a dilution series. We tested it for 9 organisms. You can see that it's a paired chart. So you have the calScreener assay and the compendial assay next to each other. And it's only when you see a difference in height of the bar chart that there is a difference in between the 2 systems. We have run some quite early statistical calculations, but I think it's quite significant for the state where we are. We have seen that the most probable number for both the compendial and our system is around 1.6 CFU, and they are at the same level for both systems. So that means that we have an LOD of around 1.6, which is, I think, a very good LOD, and also an expected number from both the compendial assay and that our system is non-inferior to the compendial assay. If we then take a look at how the system is working when you have a product present and what effects the matrix has on time to detection, we will go over quite a lot of different product cases. But before we jump into that, I would like to just show you the principle of how we're measuring and how we're detecting, and how, for instance, a T-cell culture affects the detection of a microorganism. A very common question that we get is how can we detect the metabolic activity or the heat generation from microorganisms, in this case, like you're seeing here, a Staph aureus, in the presence of a living product such as T cells that also generate heat. And this slide is divided in three parts. You can see growth media only, in this case FDM, in the beige color. In the brown color, you can see FDM and urcat cells, 10 to the 6 cells per milliliter. And then you can see the combination of urcat cells and Staph aureus in FDM. And it's below 5 CFU with Staph aureus, just to illustrate the kind of detection time of the system. And what you can see when there's no microorganism present in the sample, you can see a stable signal that is slightly decreasing for the FDM in combination with the T cells. And this is because that the growth mediated cells does not promote metabolic activity for the product itself. However, when you add a microorganism to the sample, the growth media promotes the metabolic activity and, of course, the biomass formation of the microorganism. And you can see an almost exponential increase in metabolic activity. And this is significant for all the microorganisms that we looked at. The height of the metabolic activity, of course, varies. You saw it was lower for Micrococcus. It can look different for E. coli, where you can have several peaks, aerobic and fermentive peaks, or as in this case for Staph aureus, that you have one major peak that continues for a very long time, where you have a sustained metabolic activity. I will show you 2 slides of different microorganisms and testing of 10 different products that we've done in-house. And I will not go through each product, but I think it's an illustration of how the product affects the time to detection compared when you look at the time to detection for the same microorganisms in the absence of product. So the purples are samples and the gray dots are just microorganisms grown in the growth media. And as one can see here, that in most cases for Aspergillus brasiliensis and Candida albicans, there is actually a small effect, or in some cases a larger effect of the sample on time to detection. And in some cases, for some products that you see here, there is no positive control as it looks at here, but that's not the case. It's just that there are overlapping samples and they get the same time to detection. But for some samples, you can see there is a difference in between time to detection for the sample and for when you're growing it without sample. However, for both of these organisms, it's not affecting the time to detection for the whole assay. So time to negative still remains below 72 hours, even though we're testing a lot of different products at a lot of different setups, both when it comes to the matrix, cryopreservance, and also the amount of cells in the sample, for instance. When you look at C. acnes, that is the rate-limiting organism, you can see that basically in this case, there's no or very little effect in between the product and the positive control. Even here in 2 cases, the positive control is slower. This is just an effect of a different amount of CFU. So if you aim for below 10 CFU, as it is in all of these samples, the distribution of cells can be quite large in between some of these samples. And that means if you have 1 or 2 more cells for C. acnes, since they have a generation time of 4 to 5 hours, that can have a quite large random effect. But for the rate-limiting organism C. acnes, we actually see almost no negative effect of adding the sample to the system. Same pattern here when you look at 3 other microorganisms that are much more fast growing. They're not rate limiting for the system, but we don't see any pronounced inhibition of the product in our system. If we then wrap it up and look a little bit on some of the points that I tried to convey here today, and that is that we can offer a very rapid phenotypic sterility testing system. We can even, on challenging organisms, give you results within 3 days in the presence of products. The system itself are very much geared to detect slow growers, such as in C. acnes, and we can give a very reliable result that we have tested on a lot of different products. So we know that the product itself are not inhibiting our most slow-growing organisms. We are conducting our primary validation. We expect to finish now during the next couple of months. So that data will be available for you to look at and also maybe give you a better perspective on how our system is performing. We are also offering proof of concept studies and testing, so you can explore the system without actually having the system in your lab or having to set up a lot of quite kind of larger testing screens or something like that. So there is an availability of sending your sample to us. We test it on your product, and you can evaluate how it works with your product, and we can discuss the results from there. Last slide, I just wanted to thank the team that actually produced all the data that I sit here and present here today. Fantastic team that is very dedicated and working very hard. Some of you might have met, or you may have the pleasure of meeting if you send samples here or start working with us. So thank you very much for your attention. And if you have any questions, please let's start a discussion.
Thank you very much, Wilhelm. Yes, and so now you are welcome to send in questions to the Q&A function. I can start off with one question from me. You mentioned a little bit about the proof of concept feasibility testing there. What is the typical scope in the studies we do, organisms we test, and roughly how long time the study takes?
Yeah, thank you for your question. Well, usually we test 1 to 2 to 3 products somewhere there, depending, of course, on what you want to test. We usually have about 6 to 9 microorganisms. You can either run the 6 USP organism or include the full list of the European one, the compendial ones there. Or if you have an organism of concern, a facility isolate or something like that, we can also easily include it. Usually, from when you ship the sample, it takes 2 to 3 weeks before we have run your samples, and we can deliver a report to you that we say how your product is performing in our system.
Thank you. Another one on publications and external presentations. What have there been so far on the instrument and assay, and are there any upcoming ones you want to highlight?
So there's quite a lot of publications that are upcoming, but one that I can mention because it's been presented quite a lot and was even supposed to be presented at the PDA now in October, but due to the American governmental shutdown, it couldn't be presented. Is a publication from NIST where they done an intra-laboratory study in between different labs with different technologies on TSEMs, the same type of TSEM sample that was sent to all the labs. It's done at two different challenge levels with 4 microorganisms, and I think it will give a very interesting overview of how different technologies can cope with these type of quite tough to measure samples, such as T-cell samples are. There's also been a couple of presentations during 2025. Two to mention is one was at the European Microbiology Conference in Vienna this spring, where the technology was presented and discussed, and it was presented by Roche. And then we had also a presentation by J&J at PharmaLab of the technology and how they have applied it and tested it.
Thank you. I got one question here in the chat on the test volume. So if 1 milliliter of sample is to be tested, and it's comments on that regarding split up of the samples or then in general on the sample volume to measure it.
Yeah. So if one wants to split 1 milliliter, one can either do it by a risk-based option that you say, okay, you actually do 6 vials and you use 1.2 milliliter product because you don't want to discriminate between TSP 25 or TSP 35 or FDM. There are also other ways of doing it, but then one needs to motivate why you put, for instance, more at TSP 25 than you do at TSP 35 and FDM 35. But you could see it that you split it up in 4 vials and you run it that way.
Yes. Also a question here on, you mentioned that the instrument reads every 5 seconds. Is this correct? And at what time does the instrument use the data to determine a positive or negative result?
So a negative result can first be set after the time to negative that you have with your product. So that's, of course, in our setting here, we said that we can give you results within 72 hours. There could be products where you can't do that, where you have to extend the incubation time. And with your slowest growing organism and the buffer time that you had to add, the 3.3 generations, there you will set your time to negative or when you stop the assay. A positive signal is whenever you get enough metabolic activity to trigger a positive signal that you have crossed our thresholds for detection. Then you will both on the screen get a message that says that this position is positive, the sample is potentially contaminated.
Hope that answers it, yeah. Just feel free to ask follow-up questions if there are, of course. Regarding the safety margin and time to detection, has Symcel tested cell therapy products exceeded the claimed 3-day time to detection when considered the safety margin?
So at 10 CFUs for C. acnes, or below 10 CFU as we do the testing of product, we have been able to, for all products tested so far, have a detection time below 72 hours. Then when you add the safety margin, then you, as you saw with the data and the ATCC strain, we are also below 72 hours. So far we have not had a problem with that.
On regulatory aspects, anything you mentioned a little bit there on the regulatory timeline, anything more to say on that implementation timeline for regulatory, and if there's any feedback from FDA or EMA that you want to mention?
So there's been feedback via customers from both EMA and FDA. I can't go into specific details, but basically the message from both FDA and EMA is that they see no problems with the method more than that you need to do a full method validation in order to confirm the application of the technology on your product. We are also planning to present our own primary validation for FDA, the CAT-SIBR program, in order to get feedback on it and also showcase how our data is looking like.
Okay. There was a question on the volume there. Say there is a little bit even larger volume than 1 milliliter. Any more points to mention there on volume and the system?
Yes, I think it's the system is applicable up to a certain volume. So I think at some case, at some point, it's not applicable any longer if you have to use too many vials. But covering up to 3 or 3.5 milliliter, I think is doable. How you split up a sample. If you start doing more, it becomes too many vials. Of course, it's a risk evaluation from the customer themselves or the user of the system, but I think somewhere it stopped making sense if you have too many vials.
Another question here. Have you tried testing DPs with higher T cell concentration than 10 to 6 milliliters? If yes, have you seen an effect on the robustness and sensitivity of the method? And is there pre-treatment need for DPs containing antibiotics?
We have tested up to 10 to the 8 cells. There can be effects from, all products can have specific effects on specific microorganisms. There can be a delay the higher up you go with cells in some cases for then that specific effect on microorganisms. When it comes to adding antibiotics to the media, that is problematic in the current setup because we don't have a system where we have chelators or anything handling the antibiotics. And it's very hard to dilute yourself out of an antibiotic, so I wouldn't recommend that. What can be done is that one could add something such as a penicillinase or something to break down the antibiotic, but it really depends on what antibiotics you have in the sample and if we can tailor the assay in order to deal with it. So it's very much on a case-to-case basis how we can deal with antibiotics presence in the sample.
Thank you. Those were the questions we have. Let's see if there's any more coming in. If not, you're of course welcome to send the questions to Wilhelm or myself later on. Is there anything more you want to sum up with, Wilhelm?
I think please reach out if you're interested in more information. We can discuss details in more depth. And also, if you want to send samples here to get a better view of the system, we're very open for that. So you get a better understanding of actually how the system works on your product.
Thank you. And thank you, everyone, for joining in. Wish you all a good day. Thank you.
Thank you.